Dr. Ethan Dmitrovski, director del Frederick National Laboratory for Cancer Research y presidente de Leidos Biomedical Research Incorporated. Dirige el Laboratorio Nacional Frederick para la investigación del cáncer. Sus resultados ayudaron al establecimiento de la terapia de diferenciación basada en retinoides para el tratamiento de la leucemia mieloide aguda y al desarrollo de la prueba de diagnóstico de esta leucemia, además del monitoreo de enfermedad mínima residual mediante un ensayo de RT-PCR. El Dr. Dmitrovski descubrió un nuevo mecanismo de destrucción de proteínas que causa la degradación del producto de la translocación dominante negativa de esta leucemia, PML-RAR-alfa. Y después de esta increíble semblanza, recibimos con un gran aplauso al Dr. Ethan Dmitrovski.
Good morning. I just wanted to add my thanks to Dr. Herrera for the privilege of joining you today. You know, it was really hard for me to come here today because my flight was canceled, but I would never miss the opportunity to be here with you. It's just such a thrill to visit this beautiful city and to be with this wonderful organization. Today I'm going to tell you a story. It's a story that has taken me several decades to develop. All of us as scientists get to ask questions about the natural world, and sometimes those questions lead us to answers that lead to ever more interesting questions. And you'll hear examples of questions that we posed in my own laboratory that have brought us to today to a new way to combat aneuploid cancers, which is a hallmark of cancer. So I'm going to tell you a story that began in the successful treatment of a rare leukemia that is called acute promyelocytic leukemia. Work that we had undertaken in that disease has led to the FDA approval of a drug called all-trans retinoic acid that has changed the natural history of that disease. Unexpectedly, we found that in that treatment, there was a new way to combat lung cancer, the most common cause of cancer death in the United States for both men and women. We now have the ability to target a hallmark of cancer, aneuploidy, chromosome instability, and I'm going to tell you about a new pharmacology, several new agents that can eradicate aneuploid cancers, and I hope this will be examples of interesting questions to pose in the future. Why? Because this pathway that I've described can be activated in cancers of patients. A quick primer, a quick story to tell is a story about vitamin A, which is essential for vision and for fertility. It also has effects on developing cells, but I'm only going to talk to you about the effects it has in cancer therapy and give you an example of one of the earliest successful cases of targeted therapy in this disease that I referred to, this leukemia APL. The drug all-trans retinoic acid works through nuclear receptors, and here's a quick summary of that signaling cascade. This series of nuclear receptors, proteins that reside in the nucleus, include three family members of the retinoic acid signaling pathway called alpha, beta, and gamma. All-trans retinoic acid, which has been FDA approved for this rare leukemia, activates all three of these nuclear receptors. In contrast, there's a heterodimeric partner of these nuclear receptors called the retinoid X receptors. It was called X because we didn't know the ligand. We now know that we can make drugs to this pathway and activate or repress this pathway. One example of an FDA-approved drug is called bexarotene that I'll talk about. These two pathways come together, sit on DNA, and signal changes in gene expression that lead to the biological and clinical effects that I'm going to talk about today. I'm going to show you one slide from a paper we published many years ago now in the New England Journal of Medicine. Here is an image of an APL cell in the peripheral blood of a patient diagnosed with this rare leukemia. After just 2 weeks of oral therapy, you can see there's a dramatic change in the appearance of these cells. And you need not be a pathologist to see that the cells on your right look like polymorphonuclear cells. But they're not. They're actually leukemic cells that have matured This was the first successful example of what is now called differentiation therapy. Unexpectedly and quite unbelievably, when we asked whether retinoic acid could mature these cells in patients, we uncovered that only patients who had a rearrangement of the retinoic acid receptor alpha responded to this therapy. Indeed, in a separate publication in the journal Cell, we cloned this receptor and called it PML-RO-alpha, probably an oncogenic protein you've heard about. And because I'm a physician scientist, my laboratory patented the genetic test for this disease. We sold that patent to Roche, and now we have a genetic surrogate marker for response. Whenever you have this rearrangement, 100% of patients respond to retinoic acid. When we began this work, about 20% of patients are cured, and now it's essentially a solved clinical problem with over 95% of patients cured. There were 3 or 4 groups in the world who studied this. Mine was one of them. And all of us share this incredible sense of fulfillment that work that we conducted in the laboratory actually changed the natural history of this disease. But with this story being told, we wondered, is there anything we could take from this story that might apply to more common cancers? Only in the United States, about 2,500 people a year are affected with this cancer. And what we found was that retinoic acid was, quite unexpectedly, inducing the destruction of the oncogenic protein that we had uncovered called PML-RO-alpha. And we wanted to ask the question whether we could take this drug-induced protein destabilization pathway into a more common cancer, and we were thinking about studying lung cancer at this point. Why lung cancer? Here are the United States statistics of incidence and mortality, and you can see quite unbelievably that how in the red line, both for men and women, lung cancer is the most common cause of cancer death. For women, breast cancer, as you know, is more frequent, but sadly more women pass away from lung cancer. Once lung cancer is disseminated, it's essentially incurable. And we were investigating whether we could find a mechanism, a way to use the very drug that we had used in APL to work as a cancer preventive agent. And we conducted an experiment that we published in the Proceedings of the National Academy of Sciences, where we added the very carcinogens individually that cause lung cancer, either cigarette smoke condensate or the carcinogen nitrosamine NNK. And we could create benign cells, we could create those cells and make them malignant. But when we treated with retinoic acid, we found that we could actually keep the benign cells in a benign state. And we wanted to ask the question how this can occur. And what we found, quite unexpectedly, when we asked this question, is that both retinoids, all-trans retinoic acid, and rexinoids, those that activate the RXR pathway, will induce the destruction of G1 cell cycle proteins like cyclin D1 and cyclin E. The consequence of this is that causes checkpoint arrest, the repair of genomic DNA damage, and we found that the epidermal growth factor receptor tyrosine kinase inhibitors that you're all familiar with did not activate this destruction pathway, but would, through anti-mitogenic pathway, repress the same species. Here's our original publication, PNAS, where we showed this destruction pathway. If you look to the left of this image, you can see that we have synthesized cyclin D1 protein and applied to that synthesized in vitro assay extracts from cells that were previously treated with retinoic acid, and you can see we could destroy the cyclin D1 protein. Over many years, we uncovered that the second retinoic acid receptor, called retinoic acid receptor beta, was transmitting this signal. But when you smoke, the very first thing that happens is you silence, through methylation mechanisms, the retinoic acid receptor beta, so this drug can never work in patients. And clinical trials actually confirmed that all trans retinoic acid does not work. Why? Because the retinoic acid receptor beta is silenced by smoking. However, the rexinoid pathway, the RXR pathway, that other heterodimeric partner that I described, that pathway is preserved. And what we found was that the retinoic acid receptor beta pathway actually had a critical isoform that we cloned and called the RO beta 1 prime. It too was silenced. If you look to the right, you can see in this western blot there is a polyubiquitinated cyclin D1 that only occurs when we capture it with a proteasome inhibitor. We've elucidated the mechanism for this, and it involves a phosphorylation site of a threonine residue on position 286. And if you transverse just one residue, you prevent this polyubiquitination, which is what triggers the destruction. So because I'm a physician scientist, we wanted to move this science from the laboratory into the clinic, and we conducted 5 clinical trials over the course of a decade, where we actually demonstrated that the very pathway I've just described to you is activated in human cancers, lung cancer patients. And quite unexpectedly, we saw a survival advantage, and what we found was that, unbelievably to us, the KRAS oncogene, the oncoprotein that has been a very elusive oncoprotein that we can't target, you've heard that from my colleagues at Frederick National Laboratory, we were able actually to treat patients with mutant KRAS. We saw advantages of survival, and we conducted so-called phase 0 studies, proof of concept through phase 2. And right now we are using a next-generation rexinoid, which we think will be superior to bexarotene that's FDA approved, and we hope actually to lead to FDA approval for that drug over the next few years. But this led us to a really interesting question, which was, what was the mechanism involved in these responses? And here are biopsies post-treatment versus pre-treatment from the very trials that I showed you a moment ago. And you can see, if you look from the left to the right, there's a dramatic reduction of cyclin D1 protein, total epidermal growth factor receptor expression, as well as phosphospecific epidermal growth factor receptor expression, thus validating the mechanism in human cancers that I described to you a few moments ago. But then we found something that we never expected to see. There was this wave of necrosis that in these treated tumors that was preceded by a wave of inflammation. Science is interesting because you get to ask questions, answer them, and then hopefully lead to ever more interesting questions. So we sought to investigate why this inflammatory response was uncovered, and in a series of publications in Cancer Discovery, Journal of Experimental Medicine, Blood and Cancer Research, our group and others found that retinoic acid was the morphogen for T regulatory cells. So this has very broad implications for cancer therapeutics that I'm not going to talk about today, but hopefully when I come back in the future I can tell you what we're doing to investigate how you can manipulate, monitor, regulate the immune response to checkpoint blockade, an area of active research in my laboratory right now. Rather, I wanted to tell you other questions that we posed that brought us into interesting new areas of work. But first, I'd like to show you the validation of these clinical trials, and you need not be a radiologist to see that that CT scan on the left is quite abnormal. If you look at the arrow, it's pointing to the potential space, which we call a pleural effusion, which is space between the visceral and parietal layers of the lung covering, the viscera, and that's filled with fluid. And then the second arrow actually points to lung cancer in the substance of the lung. After 3 months of therapy, you can see a dramatic improvement in the CAT scan of the patient treated with this new therapy that I've described. This patient was a non-smoker, never smoked in his life. He was a physician, and when we met him, he was bedbound, and we thought he would pass away in just a few weeks, but he actually ended up living for 18 months with metastatic lung cancer. Here's where the story becomes quite interesting. We sequenced the patient's tumor, and we found we were looking to see whether there was an activated mutation of the epidermal growth factor receptor, which would predict response to this therapy, but we actually found the wild-type sequence. So in this new therapy, what we have done is we've broadened the activity over other drugs that have been used in the past. And remember I told you that there was responses even in patients with KRAS mutation. Before I go and tell you the questions we've posed about that topic, we undertook over the course of about a decade to understand how this destruction pathway that I told you about was occurring. And it occurs in part through the classic ubiquitination pathway where you activate direct target genes that are involved in retinoic acid signaling, such as the multiple species present in the ubiquitination pathway. And we published this in a series of publications that I've cited below. But we asked the question, could there be yet another destruction pathway that was hidden by the first one? And here's where the story becomes ever more exciting to us. We've uncovered yet a new destruction pathway that involves a deubiquitinase called USP18. Unlike classical deubiquitinases that are somewhat promiscuous in their substrates, USP18 is a specific deubiquitinase that only removes a ubiquitin-like molecule called ISG15. Over a series of publications, we've shown that we can destabilize oncoproteins like KRAS, like PML-R-alpha, like cyclin D1, simply by manipulating the levels of the deubiquitinase. So this then leads to a question, could it be that this is an attractive therapeutic target? We believe it is. And we have a program underway in my laboratory to find drugs to this pathway. And we're making progress in that area. In order to convince us that we were actually looking at a vital rate-limiting oncogenic pathway, we decided to engineer a mouse that had deregulated either the cyclin D1 pathway through its associated kinase or the cyclin E pathway through cyclin D2. And we published a paper in the Proceedings of the National Academy of Sciences showing that we could actually recreate lung cancer simply by overexpressing cyclin E mice, and we've affectionately called this mouse our CEO mouse. This mouse develops lung cancers quite briskly, and we then engineered these cells under two states, one with a proteasome degradation resistant cyclin E species, the other wild type. And when you destabilize the destruction pathway, you actually get metastatic cancers of the lung and multiple lung cancers. We harvested cells from the mouse because they're syngeneic. We thought we could develop a model system that could quickly be used to analyze candidate drugs for lung cancer therapy. We've patented this mouse and we make it available to the scientific community. And we found that we could actually hone these cells back into genetically identical FEB mice. And then rather than taking 3 to 4 months for cancers to form, we now have a model where cancers form in 2 weeks. So all the clinical trials that we're doing now, we pre-screen with this syngeneic mouse model, and it is a great way to funnel agents into a higher likelihood of working in the cancer clinic. So I've told you that this target is rate limiting, but there's a very interesting question that came to our mind that I wanted to tell you about, and that is it possible that human cancer cells that frequently overexpress cyclin E or its associated kinase CDK2, are they addicted? Are they dependent on the kinase? And so we asked this question using a first-generation CDK2 inhibitor that's called Saliciclib. And here's where asking a simple question could lead to ever more interesting questions. And what we found was a new pathway for eradicating aneuploid cancers. And I'll just ask our colleagues if they could drive this video for me, please. If you could just drive the video. Let me see if I can get it to drive. Nope. Would you be able to just drive that? So I'm going to show you, if we can run this video, I'm going to show you the fate of an aneuploid cell that is, so I'm going to tell you a story about what you're supposed to see. Oh, good. So these are 3 cells. One is outside the focal plane. Now you're going to see the fate of 2 cells within the focal plane. These are aneuploid cells, chromosomally unstable, that have been treated with a first-generation CDK2 inhibitor. And what you can see is after the cells divide, the progeny cells die. You can see the first cell now becoming pycnotic in the upper portion of the middle portion of the slide, and then the second cell is dying. This death program we have called anaphase catastrophe. Why? Because it only occurs during the anaphase of cell mitosis. This is a very interesting pathway to us that I'd like to tell you the work that we've done. But before I do, I wanted to tell you the mechanism of anaphase catastrophe is the kinase inhibitors preventing the clustering of centrosomes. When I published this work, I got a call from Tak Mak at Princess Margaret, and he said, Ethan, I see the identical cells in our publication in Cancer Cell. And so we've begun a collaboration with Tak, and what we found was that unlike CDK2 inhibition, he was using a drug called polo-like kinase 4 inhibition. And you can see that centrosomes in that panel here with their drug CF1 400-945 doesn't prevent clustering of centrosomes. It leads to overduplication of centrosomes. So here we had a simple question of whether there was addiction led us to an interesting insight into centrosome biology and cancer biology. Using live cell imaging, we found that as we approach anaphase, it is anaphase catastrophe that occurs before the induction of apoptosis. So this is a rate-limiting step. Here's where the story became ever more interesting. We used a robotic screen to analyze 270 cancer cell lines linked to the Sanger genetic database. And what we found was in the upper panel, black or gray means marked or moderate growth inhibition with this CDK2 inhibitor. And when you look at the Sanger genetic database to the left, you can see that nearly every cell that was most responsive to CDK2 inhibition harbored a RAS mutation. But the 15 least sensitive cells never had a RAS mutation. Kind of a contrary result, right? Because we've all been taught that RAS mutations lead to resistance to anti-neoplastic agents. So we decided to ask the question why that was, looking at both KRAS mutant or KRAS wild-type lung cancer cells. And here's where the story became quite interesting, because if you look at the first two cells, C10 and BS2B, they are not cancerous cells. They are much less chromosomally unstable than the cancerous cells. But if you look at the cancerous cells, there is multipolarity in death, whether or not you have the RAS oncoprotein being expressed. We sought to discover the cause for that. And over about 5 years of work, we had a factor that we had affectionately called factor X. And we said this factor must preferentially exist in a cell that is activated by the KRAS oncogene that predisposes to a death program through CDK inhibition. Using a candidate gene approach, and 20 candidates we looked at, we discovered what factor X is, and it's a centrosome protein called CP110. CP110 has 10 phosphorylation sites. We mutated every site and actually uncovered the 2 critical sites that undergo phosphorylation and confer this anaphase catastrophe pathway. It's threonine 194 and serine 170. What we found was that KRAS itself regulated CP110, and that's why it's sensitized to inhibition by CDK2. Why? KRAS drove down CP110 expression. Why that led to death? It led to death because this critical centrosome protein, CP110, was repressed, and therefore clustering was inhibited ever more so. Why was this downregulation occurring, and does it have any clinical relevancy? So we acquired a database of over 550 human lung cancers and developed a paraffin-embedded assay for CP110 expression, and then linked that to RAS status or not. And as you can see by this study that we published in Cancer Research a few years ago, there's a highly significant correlation between CP110 expression and the expression of KRAS. In work that I'm not going to show you, we actually elucidated the mechanism for KRAS-driven destabilization of CP110. And what we found was that KRAS was activating cyclin F protein that led to a proteasome-dependent destabilization of CP110. So it was co-opting a destruction pathway that was activated by KRAS. It was at this point that we became aware of the drug that we were using called Saliciclib had entered into clinical trials and it was highly toxic. And over the last 2 years, we have been quite interested in looking at the next generation of CDK2 inhibitors. One is a tool compound that I'm going to talk to you about now in a story we published in the Journal of the National Cancer Institute. It's a drug called CCT68127. And as you can see in panel B, it is about 10-fold more potent than the first drug that I described. And here we used a robotic screen in the right lower panel to see whether, once again, KRAS mutant cells were responsive to CDK2 inhibition. And the robotic screen of about 100 lung cancer cell lines showed that KRAS mutation did not confer resistance to the drug. We moved this work into animal models, and we found by panel B, you can see the drug was well tolerated. There wasn't weight loss, but there was a dramatic reduction not only in tumor size, tumor volume, but also in circulating tumor cells. This drug is a tool compound. It is not optimal for formulation to deliver to patients, but there is another drug that has been used and just is finishing phase 1 studies that shows activity in phase 1 trials. It's called CYC065. CYC065 is yet another second generation, next generation CDK2 inhibitor. And this drug has shown activity in a phase 1 trial. You might ask, were there any lung cancer cases that were accrued to that trial? Unfortunately, there were no lung cancer patients that were accrued. So we have been investigating this now clinical lead compound, CYC065, and asked yet another question. Is anaphase catastrophe induction by CDK2 inhibition, is this broadly induced in aneuploid cancers? And if it's broadly induced in aneuploid cancers, does anaphase catastrophe occur whether or not there is KRAS oncoprotein expression? In this work that we've now submitted for publication, we've investigated pancreatic cancers, of which 95% harbor RAS mutations, colon cancers, which about half do, and lymphomas, which a minority of lymphomas have RAS mutations, and they all induce anaphase catastrophe. We've gone on to use PDX models, which we turn out to be quite useful tools for us to discern whether anaphase catastrophe is actually triggered in the PDXs and the patient-derived xenografts, and it's induced. So we feel this is a general mechanism. These are all aneuploid cancers. We feel this is an attractive mechanism that can be engaged in the cancer clinic to target a hallmark of cancer, which aneuploid is. So I'm going to show you another video. Before I do, I'll ask my colleagues in just in a second. We'll see this video. This is a technology called focused ion beam scanning electron microscopy. Think of it as a CT scan of a cell. And what we're now imaging is the nucleus in a CDK2-driven death program. And in green, you can see the centrioles. Something quite unexpected occurred when we conducted this study, which is still not yet published. What we found was that the centrioles did not reduplicate, and they were single centrioles. Moreover, we found that they were in a polar position. So this ultrastructural insight into centrosome duplication has not been heretofore, to our knowledge, been described, but is intimately involved in CDK2 response. And this is work that we have ongoing with a key collaborator at Frederick National Laboratory. I know you've heard from many of my colleagues about the technologies that we have. Focused ion beam scanning electron microscopy is one of those really remarkable new technologies. It's been co-opted from the engineering community that has largely used it for work in analysis of metals. It's now moved into biomedical imaging. And my colleague Kedar Narayan, who at Frederick National Laboratory is our key collaborator in this work, has just been a wonderful colleague. So I've just shown you the image analysis by FEPCM of a CDK2-driven tumor. I didn't show you the controls, but I'm going to show you the controls now. On the left-hand side, you can see the difference in treatment with a CDK2 inhibitor versus not. And what you can see is on the right, you can see that there's a single centriole, not two. And we feel this plays an instrumental role in anaphase catastrophe. I wanted to remind you that the centrosome protein that we uncovered, CP110, is a direct phosphorylation target of CDK2. That's why it was selected. So here's a protein that sits in this structure, a direct phosphorylation target, and somehow in mechanisms that are still somewhat elusive to us that we're working on, this change in phosphorylation state leads to an altered synthesis of centrioles. So how are we attacking, addressing, combating that question? Remember I said to you there were 10 phosphorylation sites, only 2 are critical for this anaphase catastrophe induction. So we're in the process, nearly done, of making phosphospecific antibodies, monoclonal antibodies to these phosphorylation sites. I'm going to stop for a second and tell you that we think this work with CP110 is actually even more intriguing than I talked to you about. And that's why, remember I told you I got a call from TAC-MAC about a new pathway that he had discovered that he thought was identical with ours, with the polo-like kinase 4 inhibitor, PLK4 inhibitor. Well, unbelievably, that kinase also targets CP110, but in an 11th phosphorylation site. So obviously we're making a phosphospecific antibody to that 11th site. Now I'm going to take you through another story that we published recently in the Proceedings of the National Academy of Sciences that talks about this other pathway. So here is why we undertook this work. It was not just the call I received from TAC-MAC, it was because PLK4 really appears to be an attractive drug target for lung cancer. Why? If you interrogate the Cancer Genome Atlas, which we did, you can see a dramatic increase in human lung cancers versus adjacent normal tissue, overexpression, dramatic overexpression of PLK4. TAC and his group at Princess Margaret has developed a highly selective PLK inhibitor called CFI 40945. This is entered into phase 1 testing, and so we're really interested in looking at how this might affect centriole reduplication. The first observation that we made in this paper I just referred to is that PLK4 inhibition promoted polyploidy, and that polyploidy led to lung cancer death. I'm just giving you the highlights of this publication. But we actually moved into syngeneic models and found that in a dose-dependent manner we could eradicate lung cancers by treatment with PLK4 inhibition. And in the right lower panel you can see these bizarre cells that are reminiscent of what we thought were anaphase catastrophe cells, but they're not. This is an entirely different death program. And this program is triggered not by inhibition of centriole clustering, but by the overduplication. And just reason along with me, you could imagine if you overduplicate the centrosomes, overduplicate this pathway, when you hit anaphase, the multipolar cells can't get through it. They tend to die at mitosis, unlike anaphase catastrophe, where the progeny, the progeny cells die. And in our publication, we thought, oh, one target, two drugs, two sites on one target, they should cooperate. So the last figure in this paper that we recently published shows that these two pathways not only are additive, but they appear to be synergistic. Why this is interesting to us is this same darn protein that we uncovered, CP110, seems to be a phosphorylation target for both pathways. Now, one can envision a hypothesis-driven clinical trial based on this, and that's obviously where we're moving. We also just published this week another paper with Tax Group. There's yet a third drug that's of interest. It's a tyrosine threonine kinase inhibitor. And I'm just going to give you the highlights of that publication, it just came out this week, is that using the Cancer Genome Atlas, TCGA, once again we found TTK was overexpressed in lung cancers versus normal lung tissues, but this seems to be a broadly active pathway. We've actually analyzed all the cancers that are interrogated in the Cancer Genome Atlas. In adenocarcinomas, it's a negative prognostic feature, and RNA in situ analysis showed that TTK is abundantly overexpressed in human lung cancers. In a series of in vitro studies, as well as in vivo syngeneic mouse model studies, we showed that the TTK inhibitor is well tolerated and causes dramatic anti-tumor effects. But this is not working through centrosome pathway, but working through yet another pathway. What this drug is doing, preferentially in aneuploid cells, is leading to chromosome instability being increased. And that seems to exceed a critical threshold that we've hypothesized to exist that leads to lethality of cancer cells. So I've told you today that we have 3 drugs, 3 pathways, each targeting a hallmark of cancer. And so now we're very excited to see where this might lead us. Why? All 3 drugs have just finished phase 1 testing. All 3 drugs are safely tolerated in patients, and they each have signals in phase 1 trials. So what have I told you today? I've told you that perhaps there's a new pharmacology that all of us can exploit, either in the laboratory, in the bench, or in the patients, in the cancer clinic. This involves targeting aneuploid cancers. We can accomplish this with CDK2 inhibition. We can accomplish this with PLK4 inhibition. We can accomplish this with TTK inhibition. 2 of these drugs seem to act on the same, we hypothesize the same centrosome protein, CP110. And we have now the mechanistic tools for us to elucidate the mechanisms involved. Aneuploidy is a previously well-described hallmark of cancer in the famous Weinberg series of hallmarks of cancer publications. I've presented this work at a meeting that Bob Weinberg was at, and he said, well, the big question is aneuploidy the cart or the horse? We're arguing it's the horse, it's driving the cart. And I got positive feedback from Bob about that. We have uncovered 3 new drugs. I'm sure there'll be others that target aneuploid cancers. But because of this unexpected mechanism of cyclin F-dependent, KRAS-dependent cyclin F destruction of the centrosome protein CP110, it opens up a tractable way for us to treat KRAS-driven cancers, like colon cancer, like pancreatic cancer, like lung cancer, et cetera. These unmet medical needs that the previous speaker spoke about. This is really what innovation discovery has to be about, touching unmet medical needs. And you know, I was reflecting on last presentation, Lewis Thomas, the very famous commentator about science and society, talked about such technologies. And he said the reason healthcare is so expensive is because we have halfway technologies. We don't have decisive technologies. The work that we did in APL that led to 95% of patients ultimately being cured is an example of a kind of therapy that changed the natural history of a lethal cancer. It's not expensive because it's oral therapy. It's not expensive because it's not halfway technology. And what our field needs is these kinds of decisive technologies. Whether targeting aneuploidy is such an approach, time will tell. We don't know enough yet to know if that's the case, but we're eager, my lab is eager to consider that possibility and continue this work. So these agents have entered phase 1 trial testing. They're now soon to start phase 2 testing, and we're excited to see what the future will bring. My role in this work is largely just to be a spokesperson for others in my lab at Frederick National Lab, colleagues at MD Anderson. George Wilding is a close colleague of mine, and I had the pleasure of working in the room next door to George. George was the talented one, I want you to know. I'm really excited about working with my colleagues at Princess Margaret, Tech Mac, Kelsey too, and really a cadre of colleagues who've been fun to work with. And you've seen some of the presentations of my colleagues at Frederick National Lab. I collaborate with so many of them. Today I've talked about the work I'm doing with KDAR. And all the science that we do is really not the work of us as an individual, it's the work of us as a team. And I've just been fortunate to have wonderful colleagues to work with in all the institutions that I've been part of. And I just want to say to Dr. Herrera, thank you so much for inviting me. This is a meeting I treasure being part of. It's just been a pleasure to be with you. Thanks for your attention.